Right, welcome to the cooking show by me, Rawdon Gamzee. You know who it is, the real chef. On today's episode, we're going to be making mammoth meatballs. And I know what you're thinking. Aren't mammoths extinct? Didn't they die 10,000 years ago? Well, you're right. They did die 10,000 years ago. But guess what? A company out in the Netherlands, I don't know what they're doing in the Netherlands, but they made and managed to synthesize and grow mammoth meat. Yes, you heard that right. Mammoth meat, the animal that died. And they were the first ones to do it and the first ones to create meat of an extinct animal. So now, let's look at how they did that and follow their recipe for getting the meat before we come back here into the kitchen and follow my recipe to make the best mammoth meatballs you'll ever have.
Okay, so to first make our delicious meatballs that's meaty, juicy, and just the right texture, we need to first synthesize or grow our mammoth meat. Just like this recipe for the perfect meatballs, growing an extinct animal's muscle protein also requires a very fine-tuned recipe. I will be referencing a recipe from our good old friends at mammothmeatball.com, who were the first ones in the world to successfully synthesize and grow muscle cells of an extinct animal. Here is a step-by-step video on how they did it for you guys wanting to try it at home, although I don't recommend doing it at home, but you're welcome to try. Anyways, so first we need to find the right gene to get that juicy, meaty taste and texture we all love. For those of you physics kid, I'm not talking about the jeans your trendy cousin buys every other weekend at Forever 21. No, I'm talking about the genes that carry the information and determine the traits in every living organism. They all perform a specific function and give detailed instructions for cells to produce the proteins needed for building blood, hair, bones, teeth, and in this case, muscle. For our mammoth meatball, we need to first identify a specific gene, a mammoth gene, that would give our dish the same traits and tastes as a woolly mammoth would have looked and tasted. This gene was identified to be mammoth myoglobin, which was found to be highly present in skeletal muscle cells. Myoglobin binds to iron and oxygen, giving that meaty taste in red meat, perfect for making a juicy meatball with just the right texture. For step 2, we need to find the specific DNA sequence that encodes for the myoglobin protein in mammoths. A DNA sequence is a combination of nucleotides in a DNA molecule and is written in the letters A, C, G, and T, short for the nucleotide bases, which are these, and I'm not going to try to pronounce them. You can try saying them in your own head. Anyways, with these letters, a long combination of code is made, and this is a language of DNA that enables us to get the right ingredient or freed mammoth myoglobin gene for our recipe. After reviewing hundreds of publicly available mammoth DNA sequences, we were finally able to locate the desired mammoth DNA sequence for this myoglobin gene. However, this sequence had several gaps that needed to be filled to successfully complete our desired DNA sequence for the mammoth meatball. This is where step 3 comes along, as we now need to complete this broken sequence using the African elephant genome. And I know what you're wondering, why an elephant and why not, let's say a chicken? The answer is quite simple. As you can see, a woolly mammoth basically is an elephant dressing up as Bigfoot, while chicken is, well, a chicken. For those of you who still don't understand or want the scientific answer, the African elephant is a woolly mammoth's closest living relative. It's like the less trendy cousin that buys super tight skinny jeans and not super trendy jeans, right? Anyways, so we can use the elephant DNA sequence and genome to help us piece together and fill in the broken mammoth DNA. Another added bonus to using elephant DNA is that the genome research on them is very extensive and with many available DNA databases to help us get the full picture of what the mammoth genome could have looked like. Once we had a complete DNA sequence for the mammoth myoglobin gene, we move on to step 4, where we now need to insert the sequence into a living target host genome. For this, we need to use a vector. No, not the type of vector you're thinking about. This type of vector is sort of like a non-toxic derivative of a virus. It sort of does the same thing as a virus in the sense that it attaches itself to a host cell to insert its own genome into the cell, but to make it a vector, all of the viral genes are removed and the vector is modified to deliver genes that you want it to deliver. In this case of the mammoth meat, the myoglobin gene and DNA sequence was synthesized and inserted into a vector, which allowed us to transfer the mammoth myoglobin gene into a sheep genome. We did this by hitting the cell with a low current, high voltage charge, enabling this vector to enter the host cell. Now, I know what you're wondering. Wait, did I just say a sheep genome, like Baba Black Sheep? Well, yes, like Baba Black Sheep, but don't worry, no sheeps were harmed. Only isolated muscle cells from them were used. Now you're probably wondering, a sheep? That looks nothing like a mammoth. Well, that's the whole reason why the sheep genome was used, since their myoglobin gene is significantly different from the mammoth myoglobin gene, and so we can be certain that our sheep cells are in fact producing large amounts of mammoth myoglobin and not failing and producing sheep genes again. Anyways, now on to step 5, the last step, which is where we multiply these cells. So, to do this, we add some essential micronutrients that help us create a tasty textured meat profile, including sugars, salts, vitamins, and amino acids. We then placed the cells inside climate-controlled cultivators, thereby creating a natural way of forming muscle tissue, just like a mammoth would have done naturally. If you're curious how long you have to do this for, it's until you have enough cells to make meat. For example, in this recipe, I'm going to use 20 billion cells. But you can double or half the recipe as much as you like. Which brings us to the final product, mammoth meat. I bet your meat looks just as delicious as mine. Anyways, back to the kitchen we go to combine even more yummy things to make our mammoth meatballs.
Alright children, now that we know how to make the meat, let's make the meatballs, eh? So, first, we got the meat in here. You can't see it, but it's there. Next, what do we add? We add the garlic. We all love garlic. Now what? Onion. Make sure it's fresh onion. Now we got ginger, the best thing on earth. It's so good for you. Now, an egg. Right from your chicken, right from your mum's farm, okay? Make sure it's from your mum's farm. If it's not, come get it from my mum. Next, we add some cinnamon. Now you're probably thinking, cinnamon? What's that for? Trust me, I'm Rawdon Gamsey. Trust me. Add some cinnamon. Next, some paprika. I don't really believe in this stuff, but my daughter likes it, so we're gonna add it anyway. Oregano, everyone's favorite. The Italians, I can hear you. Next, the king of all spices. Salt. This says olives, but I promise you it's salt. You can see it. It's not sugar. It's salt. Make sure you use salt, not sugar. Oh, we forgot something. Pepper, also a king of spice. Now we mix, guys. Make sure you mix it well. I'm watching you. Now we take the thing, chuck it in the oven, eh? If there's extra pans in there, even better. Now, we gotta leave it for a couple minutes. Close your eyes. Are you closed yet? Are they closed? Alright, time to go check on them. Time to get them back. Just look at that. Perfectly made meatballs. And that's how you do it. That's how you do it with Rawdon Gamsey.
Hi, I'm Stevette Irwin, and welcome to Get Wild with Stevette Irwin. Extinction is an issue that we are facing at an ever-increasing speed. It is estimated that by 2050, 1/2 of all species could become extinct. De-extinction, science which focuses on bringing back animals that we have lost as a result of human activity, could be the key to ensuring that our world's biodiversity does not fall to dangerously low levels. Today, I'll be taking you on a journey to discover 2 wonderful birds that have been brought back to life using these methods. Join me to discover this wonder of science and the technology behind it.
few moments later.
Oh, crikey! Look over there! A huge flock of dodo birds! Let me quickly and slowly approach them. Now, dodo birds are very curious animals. They do not have any fear of humans, and that is one of the main reasons why, in the 1600s, they went extinct. But now, as you can see, they have returned. Let me go quickly pet one. Hello there, little dodo bird. See, these are very nice and docile creatures. No reason to fear them at all. And yeah.
A few moments later.
Oh, blimey! Look over there! The pigeons, they're flying right over our heads right at this moment. Look at the beautiful, glorious migration of the passenger pigeon. Oh, I never thought I would see this in my lifetime. It's just so amazing that we can bring back this beautiful species to our forests in Ontario and the United States. Now, I'm sure you're wondering, how is it possible to bring these animals back from the dead? Well, there are 3 methods which scientists are using to make this work: backbreeding, cloning, and CRISPR genome engineering. Now, backbreeding is the simplest method as it doesn't involve DNA manipulation in laboratories, but rather a process that has been used for hundreds of years: selective breeding. Scientists choose individuals from a population of the desired extinct species' closest ancestors and breed them together in order to achieve specific phenotypic traits. They continue this process for many generations until they have hopefully recreated the extinct species. It's a bit like backwards evolution, huh? Scientists have attempted this method with several different species, including quaggas, an extinct close relative of African zebras. The issue with this method is that it is not very precise. The breeding process is quite random, and even if we are to make an animal look like its extinct relative, that is far different from actually recreating the species. It also takes many generations in order to yield results, as selective breeding is an extremely slow process, and mammals, which are most commonly associated with this method, tend to have long gestation periods. The other 2 methods involve manipulation of DNA. Luckily, we have many samples of animals that have more recently gone extinct, such as the wonderful passenger pigeon we saw earlier, which died out in 1914. In order to recreate the flock, scientists used samples from Martha, the last living passenger pigeon who died at the Cincinnati Zoo. The issue with these DNA samples is that they are old and beginning to deteriorate. This leaves the genetic code in pieces with many missing gaps. Part of the challenge for scientists using DNA manipulation techniques is restoring the DNA to a usable sample. In the past, cloning techniques, similar to those used to create the sheep Dolly, which we talked about in a previous episode, could only be used on recently extinct or currently living species. In 2003, scientists successfully brought back the Pyrenean ibex, a species which had very recently gone extinct, for all of 7 minutes before it died of a lung defect. The recent development of CRISPR gene technology, however, has allowed scientists to be able to edit DNA before beginning the cloning process. This allows them to take DNA from the much older specimens, like the passenger pigeon from the early 1900s and the dodo bird from the 1600s, and splice the fragments together with the DNA of close modern relatives in order to recreate accurate and fully complete DNA samples, which can then be inserted into nuclei and cultivated to become cells. Does this mean that one day we could have dinosaurs roaming around the Earth again? Well, unfortunately, dinosaurs are out of the question. Since they existed hundreds of millions of years ago, we don't have any active DNA samples which can be used, but perhaps that's for the better. The oldest animals we can recreate today would date back to 10,000 years ago to the last ice age during the time of the woolly mammoths. That should be exciting. Stay tuned to learn more after this short commercial break.
Sweet country Canada, but I am not trust anywhere else.
Hello and welcome back to another episode of your favorite talk show ever, The Scene. Today we are discussing de-extinction. For all those at home who don't know what it is, it's a process of bringing back already extinct species, essentially from the dead. It's a process of genetically mutating current species so that they fit the genes of previously extinct species. Some examples of these that are currently present are the dodo bird and the passenger pigeon. And we've also heard talks of bringing back mammoths just so we can make meatballs out of them. Isn't that crazy? Hooray.
Yeah, I mean, this is a really interesting topic that we're talking about today, and I'm super excited to hear what your opinion is on it, Eris.
Yes, to begin with, I just want to make it known that I am completely against this entire de-extinction program.
You're against it?
Yes, I find it absolutely bonkers that we are focusing on bringing back extinct species and not worried about protecting and conserving species that are really in danger of going extinct today.
Okay, but think about the benefits that this process could have. Like, for example, we could be helping to promote better biodiversity. It could help us to learn about how to bring back species that are on the verge of extinction. And not to mention that we could save the polar ice if we bring back mammoths. There's so many potential benefits to bringing back these species that we have accidentally caused to go extinct, and we can make up for that now.
Accidentally?
Well,
I mean, I would argue that a lot of times we didn't do it accidentally. I mean, we knew what we were doing. I mean, take for example the dodo bird. I mean, we just came onto their island and we completely took over their island of Mauritius.
Yeah, but we regret it now, and we can see, I mean, back then we didn't understand the benefits of biodiversity, how they help to promote health for our planet, how they help to ensure that we can maintain the environment that we live in.
But who's to say we won't do it again? Who's to say we've really learned our lesson, I mean, I go outside, we hear all of these anti-littering propaganda, but I go outside and I promise you, within 5 steps of me outside of the studio door, I will see at least 5 pieces of plastic and another 3 water bottles.
Okay, I mean, you do have a point there, Eris, but okay, let me give you some examples, Have you heard about what's going on in Africa with the conservation efforts to help save the northern white rhino? They had to use the same type of technology, of de-extinction technology, and they're looking to insert an embryo from a northern white rhino into the southern white rhino in order to help rebreed and bring back the population. Don't you think that that's a good idea?
I mean, sure, I guess, but should we really be worried about trying to bring back the extinct ones? I mean, you mentioned the northern white rhino, they're not extinct yet, I mean, I'm concerned with the dodo birds and the passenger pigeons. I mean, who's to say that they won't go extinct again? And more than that, who's to say that we have space to have them up and living again? I mean, there's data showing that since their extinction, I mean, already 50% of their native land and other land on this earth has been completely transformed to fit our human use. Where can we keep these when there simply is no space for them?
That is true, but at the same time, we're seeing that extinct species have the potential to help us save our planet. Have you heard about the woolly mammoths and how they grazed on grass?
The ones we're turning into meatballs now.
Oh my gosh. Okay, we will get to that topic after, but listen to me. The woolly mammoths, they grazed on grasses up in the Arctic. And that grass really helped to conserve the permafrost. It shielded the permafrost from the sun's rays and helped to prevent melting, right? And scientists are looking at this and they're thinking about, well, can we bring back the woolly mammoth to do that same job now? And would that help us to prevent the world from further deteriorating, from further deteriorating, that is a difficult word to say, into global warming and soupy mush?
I guess that is a good point, but I think we should also recognize the reason why they went extinct. I mean, the woolly mammoths went extinct for a reason, and even since their extinction, they've been constantly, there've been multiple new species that have emerged that have oftentimes simulated the role that they've had on the ecosystem. Should we worry about those? How would the woolly mammoths and all these extinct animals how do I say, how would they like adapt into their new environment with all the new species and the new climate? Should we be worried about the already existing species? I mean, you could argue that this could be a case of like alien species coming in. What's the word for it? Invasive species. You're right.
Okay, I think, you've brought some interesting points, but can't you also acknowledge that there are benefits to this technology?
I guess so, there are benefits.
Okay.
I mean, did you ever think you would see a day where Zac Efron and the dodo bird were existing? On that note, did you know that Zac Efron and his girlfriend just broke up?
Oh my god, the blonde one?
Yeah, the blonde one. Okay, wait.
That's
I need
absolutely